This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: What is a microarray? Answer: A microarray is an array consisting of thousands of DNA oligonucleotides arranged in a grid on a glass or silicon chip. Question: How are DNA or RNA samples being compared attached to the microarray? Answer: DNA or RNA samples being compared are attached to different fluorophores and hybridized to the array. Question: How does the ratio of fluorescent signal at a particular oligonucleotide reflect the relative amount of the hybridizing nucleic acid in the two samples? Answer: The ratio of fluorescent signal at a particular oligonucleotide reflects the relative amount of the hybridizing nucleic acid in the two samples. Question: What is the main use of microarrays? Answer: Microarrays are mainly used to compare the relative transcription of genes in two RNA samples. Question: What other type of analysis can microarrays be used for? Answer: Microarrays can detect single nucleotide polymorphisms and copy number variants for genotyping, clinical genetic testing, forensic analysis, and cancer mutation and genetic linkage analysis when DNA is used. Question: How do microarrays detect single nucleotide polymorphisms? Answer: Microarrays detect SNPs by comparing the relative amount of the hybridizing nucleic acid in the two samples. What is the use of copy number variants in microarrays? Answer: CNVs can be used in microarrays for genotyping, clinical genetic testing, forensic analysis, and cancer mutation and genetic linkage analysis when DNA is used. Question: How does microarray technology aid in cancer research? Answer: Microarrays can be used in cancer research to detect genetic mutations and variations that may be associated with the development of cancer. Question: In what fields is microarray technology used? Answer: Microarray technology is used in fields such as genetics, genomics, molecular biology, and biotechnology. It is also commonly used in medical research and clinical genetic testing. Question: What are the advantages of using microarray technology? Answer: The advantages of using microarray technology include high throughput, cost effectiveness, and the ability to analyze multiple samples simultaneously. It also allows for the detection of genetic variations and mutations that may not be detectable by other methods. Question: How is the microarray chip prepared? Answer: Microarray chips are prepared by printing or spotting DNA or RNA oligonucleotides onto a glass or silicon substrate in a specific pattern or grid. Question: How do researchers attach DNA or RNA samples to the microarray chip? Answer: Researchers attach DNA or RNA samples to the microarray chip by hybridizing the samples with the oligonucleotides on the chip, usually using fluorescent labels to track the samples. Question: How are the results of a microarray experiment analyzed? Answer: The results of a microarray experiment are analyzed by comparing the fluorescent signals from the samples to the oligonucleotides on the chip. The ratio of fluorescent signals can reveal the relative amounts of the hybridizing nucleic acid in the samples. Question: How can microarray technology be used in clinical genetic testing? Answer: Microarray technology can be used in clinical genetic testing to detect genetic variations and mutations associated with inherited diseases or disorders. It can also be used to analyze chromosomal abnormalities and copy number variations. Question: What are some limitations of microarray technology? Answer: Some limitations of microarray technology include its complexity, the need for high-quality samples, and the potential for experimental variability. Additionally, it is not capable of detecting structural variations such as inversions, duplications, deletions, or translocations.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the first aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.